This recorded session is dedicated for module number 5, Microorganisms in the Environment. This will serve as our pre-laboratory discussion. So um, I will be detailing how you guys will perform module number 5. No? So for starting module number 5, I have to make an official announcement that with module number 5, we will start submitting worksheet outputs in groups. So I have assigned groupings per or for every module that we're going to tackle, different groupings will be observed. So for this module, for the current module, I have already uploaded the list of group members per group. Please take a look at the laboratory worksheet. What do you call that? Laboratory worksheet submission part. Okay, I have divided your class into 4 groups. So you have to take note of who your group members are. So before we begin with our pre-lab, no, let's have a quick recap of what we have learned so far. So last week, we are able to carry out our fourth or our third activity, which is on culture media preparation. So during the pre-lab, I talked about the different types of culture media based on components and based on form. So based on components, we have chemically defined media in which the component ingredients are all defined compounds. While for chemically complex media, these culture media are those with ingredients that are not fully defined, not fully, cannot fully be identified based on its contents. As an example, we have glucose minimal salt agar for chemically defined medium, and we have nutrient agar as an example for chemically complex medium. Um, Aside from the categorization based on components, we also have culture media based on form. So we have liquid, semi-solid, or semi-liquid, no? Then the solid type or solid forms. So for solid, we have different preparations. You can prepare test tube butts that are normally 8 ml in volume, agar plates that contain 20 ml in volume. You also have test tube slants that have 8 ml in volume. For liquid media, you can normally we prepare liquid media in small batches. So tubes lang natin sila nilalagay. The volume most of the time is 8 ml or less based on whatever you need. No? Okay, so different forms of media will be important based on the function or based on the task that you need to do. If you want to grow bacteria on a solid medium, of course you need to have a solid medium. If you want to observe their colony characteristics, you have to grow them in solid medium. So you use either a butt, a plate, or a slant. Now, if you want to observe motility, no? A good type of medium is semi-solid medium. No? Then for liquid media, if you want to increase the number of the cells of your pure culture, then you grow them in broth. It's easier. Most bacteria will or most microbes will find it easier to actually suspend themselves, no? In a liquid matrix where nutrient is very very rich. The nutrient concentration is very high. Okay, so aside from the type of media based on component and based on form, we also made a quick discussion about how do you do media preparation. No? So as I've discussed during the pre-lab, where um, we mentioned about different we mentioned the total sequence, no? O yung ano natin yung I mentioned about the sequential steps needed in order for you to prepare your culture medium. So For media preparation, I think I have discussed lyophilized medium and compounded. No? So there are instances in which you will need to compound your culture medium, meaning you have to create it from scratch. So no, this is compounding media normally is applicable for chemically defined types. Say you have your ingredients identified, then you measure specific weights or volumes of each, and then you combine them in the specified volume of distilled water, or whatever your solvent is. And then um, in our lab, actually, in face to face, no, pag ginawa natin to in the usual setup, our culture medium are normally lyophilized, meaning they're already powdered. Then you just have to get a total volume or a total weight of your lyophilized medium, and then combine it with a specific volume of water, a specific volume of solvent that you need to prepare the total preparation. Now, in the process, diba, we follow standard formulas for each of the for the different ingredients and the total medium for the case of lyophilized medium. No. So you just do proper ratio and proportion in order to identify what amounts do you need, no, total lyophilized medium you need, or like if it's broken down into several ingredients, you can identify what specific weight is needed. If, let's say, you're asked to prepare 500 milliliters of your culture medium, then the standard formula that we have in practice are always prepared for 1 liter, no? For 1 liter or 1,000 milliliters of our culture medium. So you just have to do ratio and proportion to identify uh, components of the medium, or for the case of compounding, or kung may lyophilase naman na prepare mo, so total siya, no? Total weight. Okay? And then lastly, I've mentioned about media sterilization. It's very important that we sterilize our prepared medium because we want to start with a contamination-free or contaminant-free culture medium. No? The idea is our culture medium will provide only the nutrients needed without having contaminants that can inhibit or do something to the microbial organisms that you are trying to grow. Then you observe also how different media sterilization is, or how heat is important, no, for media sterilization. So standard sterilization in the lab is via the autoclave, where you have your parameters are 121 degrees Celsius, a pressure of 15 pounds per square inch, and a sterilization time of 15 minutes. Then for oven, we have in the virtual laboratory, you will what do you call this? You will discover how different oven sterilization is compared to autoclave sterilization. There are obvious differences, or there will be expected differences in the quality of the medium that is sterilized via the oven and via the autoclave. Okay? So much about the recap, no, for module number 4. We'll begin our discussion now with module number 5, the microorganisms in the environment. At the end of this activity, you, the students, the performers of the virtual simulation, are expected to first demonstrate presence of microbes from the environment through basic detection and characterization techniques. Essentially, appreciate how it is done, no? Appreciate how the demonstration was done. I have provided videos. I think I only found 2 relevant videos, and then a photo documentation so that you can visualize what's being done, no? The procedure in the module. Then lastly, also be able to examine and characterize colonies of bacterial cultures from the plate samples. So for this one, what I designed the module to be like is, or I ensured that the TLO13 is somehow parang nagawa nyo rin siya ng face-to-face. No? So later I will explain how we can really achieve that topic learning outcome, that activity objective. All right, so microbes and infections. We have a by now, based on your lecture and your progress in our discussion, no? In microbiology course in general. I hope that you're appreciating how diverse microbes are. No, we only do not have bacteria or fungi as our representative microbial categories or microbial groupings. We also have our eukaryotes like protozoans like amoeba and some what do you call this, some other non other representatives from photosynthetic lineages of unicellular protozoans, no? And then we have biological entities, no? Semi-living entities. We have viruses. So all of these are representative members of microbial groups that can cause infection to humans, to animals, and to plants. No? So um, uh, time immemorial, we've discovered, or the human race discovered that some infections can be attributed to I don't know, some microbial groups or specific microbial species. Like wound infections, we can uh, associate with skin bacteria. Acne is a very nice example of a pathology that's caused by a microbe, no? Propionibacterium acnes. Propionibacterium acnes is the causative agent of acne. What else? We also have, in certain instances, we have foodborne bacteria, okay, foodborne microbes. If you've eaten food material that's not properly cooked, or the number of microbes, the microbial cells, the microbial load of the food is unusually high, you know, lethally high, then you will definitely suffer from an infection, from a pathology. Okay, so there's a clear causative relationship, no, cause and effect relationship between microbes and infections. Okay, so um, aside from from what we know that, or from what we've opened the discussion with, in which we um, mentioned about microbes causing infections. Okay, microbes can also carry out a diverse set of activities or processes that are not only important to humans but also to the entire biosphere. No? Microbes are capable of are very heavily involved in nutrient cycling, nutrient storage, in terrestrial and aquatic ecosystems. Also, some microbes are associated to bioremediation. Those that can, as a good example, those microbes that can degrade oil or degrade plastic, they're involved in that in bioremediation. Then also microbes that are participating in the production or microbes that undergo fermentation, and then yung ano nila, yung process nila of fermentation is harnessed by humans in order to create fermented products, and a lot more aside from nutrient cycling, food production, bioremediation. There are a lot of other things that microbes do. Okay, because of this diverse set of capacities, it's undeniable that we can see them everywhere. We can isolate different groups of microbes virtually everywhere in this planet. As they are widely distributed in our environment, they can be lingering in the air that we breathe, in the food that we eat. Like if we are in the classroom setup, probably I will be pinpointing what areas of the classroom are most likely possessing a number, no, great distribution or high bacterial numbers or microbial numbers, and of course in our body as well. We carry a lot of microbes in our body, our normal human microbiota. Now, From the environment, no, the idea of realizing, or the idea of associating microbes in the environment, or considering that, or not associating, but the idea of thinking that microbes are present in the environment, no, it's not that really very old, no. If you will remember this, or if you've encountered this illustration, no, probably you will be, what I'm going to discuss next. This is actually the very famous um, experiment by Louis Pasteur, in order in his attempt to prove that microbes from the environment can contaminate, or can microbes from the environment, like the air, a specific environment, can contaminate our let's say, for example, a broth, no, a nutrient rich broth. Okay, so what he did, he bent the neck of this long flask, so para siyang naging swan like in shape, no? Actually, this is yung swan flask experiment niya, ni Louis Pasteur. What he did is, initially, he, while it's not bent, no, what happened is, or what the sequence of events is, he boiled the broth so that to kill any other present microbes in it. Then eventually, after having a sterilized broth, now he, what do you call this? He bent the neck of the glass in order to, prevent the entry of microbes from the environment. Then afterwards, what happened next is, um, uh, what happened next is, as expected, no aerosolized microbe from the air was able to reach the broth. So hindi siya nag-spoil. Okay, so providing us the idea, or proving us the idea that indeed from the air there are microbes. Once that they land a nutrient-rich surface, they can grow there, and they can get the nutrients from that source. But if you prevent them from accessing that nutrient source, of course, no, or if you virtually block these microbes, then you can, keep the sterility of your nutrient rich environment, your the nutrient rich broth. No? So the idea here that Louis Pasteur is really emphasizing is, there are microbes even in the air. There are microbes everywhere. Okay. Now, in our body, we need normal microbiota in order to keep our day-to-day functions. No? So, for example, those microbes that are found in our intestines, those gut bacteria, they aid in the digestion of the food that we eat. They produce fermentation products, enzymes, that can, help us digest the food matter, or the food organics that we have consumed. No? However, if in times where numbers of these normal microbiota, or not all the time yung normal lang, no? In some instances, there will be intruding microbiota. So in cases in which the number of these microbes grow beyond their normal population size, or if, let's say, for example, they populate in an unusual body part, let's say a skin microbe that became that was, sabihin natin na ano siya, no, napunta sa tiyan, or na transmit into any other part of the body where they were able where it was able to grow, no? If in such cases, yeah, infection or disease will definitely result, no? In our lab, in the microbiology lab, the working environment is a potential source of microbes that can contaminate the pure cultures that students are working on. And in and This contamination process, it can progress to human infections, especially if proper care is not observed during the laboratory activities. I remember showing you a generalized video on aseptic technique, diba? In microbiology lab. So I hope that by now we can, from time to time we can go back in reviewing that, and by now we're familiar with the basic aseptic technique. Okay, so if aseptic technique is not done or not executed well while you're working on or while you are working on your microbial cultures, diba? They can transfer to you, to your bodily your body clothing, your bodily parts, and you might not know it. You have taken them home with you, after you leave the laboratory, and they may cause infection if uncontrolled. Now, in this activity in module 5, our main focus is to be able to learn how we can characterize, how we can describe microbial colonies, bacterial colonies, those colonies that are isolated from the laboratory environment and from the human body, how we can characterize them. Why is this task very important? The thing is, some virulent microbial or bacterial species, they manifest specific specific characteristics, no, or specific morphological features that are distinguishable from non-virulent or less virulent counterparts. As an example, I have here two photos. No? So this is blood agar, and then the species growing in them are different. For this picture, this is a colony of Bacillus anthracis, which is a causative agent of anthrax. So you see how the colony looks like. Later, once that you've encountered or once that I have introduced how we can characterize them, no, you might want to go back to this slide and then try to describe the appearance of the Bacillus anthracis colony. On the other hand, this one is a different Bacillus species. This is Bacillus licheniformis, no, so a less virulent type of Bacillus. No? So you see very obvious characteristics, very different obvious characteristics. No? Now, um, we want to, of course, no, ensure that we know, or we somehow have an idea if what we are working on. Normally, if we isolate them from the environment or from the human body, definitely we don't know what they are, diba? So it's important for us to have a working knowledge on how we can characterize colony characteristics such as form, margin, elevation, surface appearance, consistency, and transparency, so that somehow we can initially identify if the bacterial or microbial colony we have isolated is a potentially disease-causing one. Okay. Again, I have to emphasize potentially disease-causing. Why? Because morphological characteristics, although helpful in some, some in identifying or discerning pathologic microbes from others, although very helpful nga ang mga colonial characteristics, it's not 100% not 100% accurate, no. The pathology or the level of pathology that or infectiousness of a microbial species will still depend on its entirety. So especially yung ano niya, no, genetic component, where we want to always emphasize that the virulence, the infectiousness of a microbial organism will always depend on the entirety of its genome. Okay? So something you have to remember, ha? Hindi ibig sabihin na, okay, this morphological characteristic indicative of pathology or potential pathology causing microbe, ano na siya, pathogenic na siya agad. Hindi siya, that's not the case. Okay, that's something I want you guys to remember, ha. Okay, so this is a golden rule, that we should follow or that we should inculcate in ourselves. Okay, let's move on. Aside from characterizing based on colonial features, we can also use different culture media in order to identify whether a colony is potentially disease causing or not. Examples of some of these growth media include blood agar, the one we saw a while ago. We can grow Borrelia and Campylobacter, some foodborne pathogens. Chocolate agar, Haemophilus, can be grown. Hectoin agar, Shigella, another gastrointestinal pathogen. Okay, so those examples of culture media, we can use them in order to identify a potentially disease-causing species. Now, with a basic knowledge of identifying potentially virulent microbial bacterial isolates, laboratory work that we are doing must always be coupled with the application of a septic technique in order to avoid infection, in order to avoid unnecessary infection. Okay? Now, I think this is an example I have to show you. So, the use of certain culture media. So, we have here this one on the top panel. It's mannitol salt agar. So, mannitol salt agar is a selective and differential media. It has high concentration of salt, 7.5% and above. So, it can only grow microbes that can tolerate that kind of salinity or that kind of salt concentration. Diba? For this case, we have Staphylococcus. Mga Staphylococcus, they can grow in saline environments, no? Highly saline environments, as you can see here. Uh, S. aureus and S. epidermidis, they show signs of growth, while Micrococcus, no growth. Now, aside from being a selective medium, mannitol salt agar is also a differential medium. It can differentiate the characteristics of epidermidis, S. epidermidis from S. aureus. You can see the difference in the coloration, no? The color of the plate. Why is that? MSA, it has a what do you call this? It has a special carbohydrate called mannitol that it put that serves as the major carbon source for if you grow microbes on the surface of an MSA plate. So it contains mannitol and what do you call that? A pH indicator. So the pH indicator it contains is phenol red. Now, for the case of Staphylococcus aureus, it's capable of fermenting or using mannitol. So in because of that capacity, uh, it produces byproducts that are what do you call this, that are sensed or that change that actually changes the pH of the whole plate. Kaya nagbago yung kulay niya. So for Staphylococcus aureus, the capacity to use mannitol is an indication or will result to a color change in the whole plate. And if we're going to compare the pathology or the pathogenicity of S. aureus and S. epidermidis, mas pathogenic ng multiple magnitude, ng multiple orders ang S. aureus. So in a way, the capacity to use specific sugar or specific carbohydrate is an indication of or a potential indication of pathogenicity. Then we also have another example. This one is blood agar, where hemolysis is observed. So hemolysis, which means lysis of red blood cells. Yeah. So we have different bacterial species that are grown in blood agar. We have Streptococcus pyogenes, showing us complete hemolysis, known as beta hemolysis. It's able to kill or make use of red blood cells totally, no? And then we also have alpha hemolysis, which is partial red blood cell hemolysis. So parang ang kulay niya instead of very clear, greenish yung appearance niya. No? Then we have gamma hemolysis where there's no lysis of red blood cells. For the case of staph epidermidis, we cannot appreciate any hemolysis happening. So, as a character, the capacity to lyse red blood cells is an indication of potential pathogenicity. And Streptococcus pyogenes is highly pathogenic yan. So, as such, that characteristic of it, where it can hemolyze red blood cells, it's an indication of potential pathogenicity. Okay? So, time for the virtual laboratory proper. I have uploaded three files. No? So, you have to download the laboratory guide, download as well the worksheet, and then there's a sample data that I prepared that you will be using to answer the worksheet. Okay? So, after you download the laboratory guide, please follow the sequence of the supplementary videos and the photo documentations. I want you to take a look at them per group so that you will understand how the virtual simulation will take place. Then, as you answer the worksheet, or before you answer the worksheet, make sure that you check your assigned groupings. Okay? Let's take a look at the materials. So, this is the laboratory guide, 7 pages. Okay. So, please check the videos, the supplementary videos. One for isolation of microbes from air, and another isolation of microbes from the ears. Please take note. Thus, I have here some photo documentations I want you to take note for microbes from faucet, microbes from working table. For floor and doorknob, parehas lang yung sequence ng procedure. You just have to, of course, swab the floor or swab the doorknob. Then for microbes naman from the human body, we have fingers, from the hair, from the mouth, and from the nose. Okay? Alright. Then we also have the worksheet. So here's the worksheet, Module 5 worksheet. Okay. Before you proceed with answering the worksheet, please make sure that you have checked our groupings here from the laboratory worksheet submission section. So we have 4 groups. Please find your respective groups and then talk to your members. Okay? Now, before you start with the worksheet, make sure that you follow this very important instruction. I want everyone to participate. Okay? So, indicate the specific contributions of each group member at the comment section of your submission. So, once you submit, indicate who did what. Okay? So, you will be although you as a group you will get 1 score for the worksheet, all right? In order to be fair, because of that that rule that I'm going to follow starting this module, make sure, guys, that everyone will do a specific activity or a specific specific part. No, everyone will contribute. Okay. Now, let's take a look at the procedure. I-visualize natin siya para mas madali na rin sa part ninyo. Okay. So for isolation of microbes from the laboratory environment, so we have initially isolation from faucet. So in the lab, what you are supposed to do is get a cotton swab, soak it or dip it on a sterile, normal saline solution, serve as your solvent. No? Then once that you've wetted the end of the cotton swab, you swab the portion of the faucet. Then you spread it on your nutrient agar plate. That's the same thing that we did for the other parts of the laboratory. For the table, for the floor, doorknob, no? Ganun din. You have your sterile normal solution, saline solution soak cotton swab. You get some contents from the floor, from the table, from the wall, from the doorknob, then swab lang dun sa nutrient agar plate. Okay? Now, for microbes from the human body, medyo specific per part, no? For fingers, ito touch mo lang siya. Technically, your unwashed hand, just touch the plate, touch the surface of your nutrient agar plate. For, what do you call this? For, swab, for hair, swab with a cotton swab that is soaked to sterile normal saline solution. Then plate lang afterwards. For mouth, okay, you may or may not swab using a sterile normal saline solution kasi wet na naman yung mouth. Unless if you're getting, let's say, bacteria from the lips or bacteria from the mouth opening, you may want to do swabbing. Okay, or you may definitely need to do swabbing kasi dry yung area na yun. Eh. So you get some of your contents and then plate it. So you'll have your mouth, your specific organisms from the mouth. Then for the nose, no, we have here ganun din. Um, since normally dry naman yung nose natin, you have to use or you have to dip the cotton swab to SNSS and then get some contents from your nose and plate it. Plate the contents on your nutrient agar plate entitled nose. No? Okay, so what's the major thing? The major task here. The major task here is to grow the cultures, grow the unknown microbes, unknown bacteria. Then afterwards, you characterize them. So you characterize them using different features. So you can characterize them based on their form, how they grow, the appearance of their, the shape of the colony normally. The margin, okay, how does the margin look like? Is it entire for circular colonies? Is it lobate for colonies that have very weird or amoeboid na shape? Is it filamentous, no, the margin? Is it curled or undulating? Then for elevation, actually elevation is something that you cannot perform in a virtual setup. You cannot assess, rather, using a virtual laboratory. Kasi paano mo nga naman titignan yung side ng colony? Diba? Ang mga, the ones that I just prepared, ano lang sila? Top pictures lang. Eh. So, we don't have side profile of the colony. So, you cannot really assessing elevation. But in face-to-face setting, very important that we are assessing this feature. Then we have surface as well, whether it's smooth, concentric, or wrinkled. No? Now, in terms of the consistency of the colony or the microbial colony, this is something also very important. Um, you'll be able to observe colonies that are very dry looking, they're not shiny. And some instances they're moist or mucoid, they're shiny. In some instances they're brittle, meaning they break if you touch them with your loop. And then in some cases they can be viscid, they can be what do you call this? They can be jelly-like in appearance, normally because of the presence of bacterial capsules or microbial capsules. No? So you're going to also assess them based on their consistency. Okay? So let's go to our worksheet. So here's our worksheet. Um, you need to pick one colony for each of the representative plate here. So I have nutrient agar plate labeled air, faucet, table, floor, doorknob, fingers, ears, hair, mouth, nose. Yeah. So, ito yung sample data natin. What I want you to do, guys, is to pick one colony from each of these plates. Or if you're not seeing a colony, then you just report na there's no observable colony. So you pick one colony. For example, let's take a look at air. Kunyari, ito yung gusto nyo i-characterize. This white one. So, you pick this, identify kung ano yung by the way, no, when you submit your worksheet, you have to submit your file of the sample data, your copy, with the indication kung ano yung pinili ninyo. You can like encircle it with a red or mark it with a different colored pen, no, or shape para ma-identify ko which colony you are describing in your worksheet. So lahat 'yan for all the what do you call this? For all the plates. So you pick one colony and then characterize it based on size, form, margin, surface, consistency, and pigmentation. You can refer to this again, okay, so that you will be able to answer. Now, for size, estimate na lang. Kasi obviously, we cannot really measure it accurately because these pictures are already blown up. Also, size, form, tsaka wala akong I didn't include any like some standard marking para ma-identify sana, ma-standard yung, ma-calibrate nyo yung size. Anyway, so size, estimate na lang. Form, margin, surface, inconsistency, you may want to, definitely estimation yan kasi you cannot check it visually only using pictures. Then pigmentation, color. So, meron ba siyang white pigmentation, yellow pigmentation, reddish pigmentation, gray pigmentation? Okay? Yeah, so you have to do that for table 1, laboratory environment, and table 2, from the worker, from the human, from human body. Okay? So that's it for our laboratory pre-lab discussion for module number 5. Okay, so okay, make sure guys that you follow yung natin groupings. Then lastly, last announcement, I will be providing the post-laboratory discussion for module 4. For module 4 na tayo, no? Tama ako. Kasi for module 3, yung staining techniques, I have already uploaded the post-lab discussion for that. Meron na siya. Paki-check na lang sa inyong in your module 3 section, module 3 part. Tapos yun nga. So module 4 and module 5, I will upload post-lab discussions, of course, after you have submitted your worksheets. Okay? All right, so that's it. Thank you for your time in the listening to this recorded lecture. So I hope that you will be able to perform the activity and learn from it. Okay? For questions, please let me know via our group chat or our SB message center.